It's a great pleasure for us to host today Ed Turner from Princeton. As many of you know, Ed made major contributions to astrophysics. As far as I'm concerned, he is the youngest person I collaborate with in spirit. Open-minded, very bright, original. Ed completed his PhD at Caltech, 1975, was it? And afterwards was a postdoc at the Institute for Advanced Study at Princeton. He spent some time here as a junior faculty, or right. And Ed actually commented to me once that there are two universities that he would entertain having positions in, Princeton and Harvard. And I would have wished to have him as a colleague, but every now and then he comes to visit, so that's also very pleasant. Ed wrote some of the early papers on gravitational lensing by galaxies, together with Jerry Ostraker and Richard Gott. And he is currently co-chairing a committee that involves the collaboration between the national observatories in Japan and Princeton University, and was also served as chair on important committees of Space Telescope Science Institute, the selection of the director,
all the way back to Bob Williams.
Bob Williams, and Aura as well, had made major contributions both to the science and the administration of astrophysics. So it's a great pleasure to welcome Ed, and today he will speak about a subject that he was working on over the past decade, astrobiology, but present an original point of view about the likelihood of life, as we will hear in a moment.
Thanks, Avi. Well, there are talks which are about very important and exciting subjects, and they're talks that have a lot of great scientific content. They're good work. They have, important calculations or results or measurements or something. And if you have both, then you have a great talk. But today you're going to get a talk of the first type. It's about, I think, one of the most interesting and important problems that in science on which people, in which there's a research community actively working. But you're not going to, at the end of this talk, probably know much more about how life arose than you did at the beginning. But I hope to give you a slightly different perspective. And part of the motivation and reason for this talk is that I've been paying attention to origin of life work by the people that do it for a while now. I originally got involved because they invited me to give some talks on exoplanets at Origin of Life conferences. And it reminded me of something from relatively early in my career as a cosmologist. When I was a graduate student at Caltech, as Avi mentioned, I learned that Big Bang cosmology had some really fundamental problems in it. One of them was the causality problem, the fact that the universe, looks the same in directions that had been out of causal contact with each other, were still out of causal contact with each other, like the microwave background, and the flatness problem, and various things. And at that time, people were aware of those problems, but cosmology was being done by averting their eyes. Everyone just averted their eyes and ignored the 800-pound gorilla in the room, the fact that these problems, that our model seemed to violate causality. People just didn't think about that. And we were more or less told to do that. And, the APJ was publishing plenty of papers based on Big Bang cosmology, just ignoring this huge problem with it until some people like, Alan Guth and Linday and Steinhardt and so on, who had very little common sense, decided to try to solve the problem, which seemed so impossible. And of course, then came inflation, and they did. I would say that in the origin of life community, there's something similar going on. There's a huge problem that they're aware of. If you talk to them about it, they get sort of testy, but they acknowledge that it's there, but people just aren't addressing it. And so I would like to address it today in a very radical way, which is ridiculous, but has some chance of being true. So let me start out by saying that reminding you a little bit about some of the properties of life that we'll be trying to address and why it's a challenge to understand the origin of life. First of all, life as a chemical system is an extraordinarily complex and rich and interconnected system. Nothing else we know is remotely close to it in complexity in terms of chemical systems, or virtually any other kind of system for that matter. It's just, as I'll be showing you in a little bit of more detail soon. The basic biochemistry of life, as you know, one of these sort of profound sounding statements that everyone likes to repeat, is the same for all organisms on the planet. You know, no matter the simplest microorganisms, the most complex and elaborate organisms that exist, perhaps ourselves, perhaps other things, in the huge diversity of terrestrial life, at the basic the way the cells actually work in their most fundamental ways is identical. It's carbon-based and water-dependent. And here's something that's important. It's all structured by polymerized macromolecules that do big things. They're polymers made out of simple monomers. And there are 4 general classes of these. So a small number of general classes, but many specific types of polymers that have very specific biological functions, sometimes extremely narrow. We're mostly just today going to be talking about 2 of the classes, the nucleic acids and the proteins. The nucleic acids are made out of monomers called nucleotides, and the proteins are made out of monomers called amino acids. Now, so what do we know about the origin of life? Well, one kind of encouraging thing is it seems to require no particularly exotic conditions or constituents. You know, as far as we can tell, there was nothing about the early Earth that was just bizarrely unusual in the universe, not like some very rare temperature or pressure or density or chemical constitution or some time series of unusual things. It looks like the conditions on the early Earth were, fairly easily understood naturally. So it doesn't look like the conditions under which life originated were extremely exotic. We don't know that, but there's no evidence that they were particularly. It appears to have happened only once in the history of life on Earth, but very quickly, quite near the beginning. I'll say a little more about that later, but in fact, the data we have are consistent with life having appeared instantaneously on Earth once the conditions on the surface of the Earth became such that life could survive. Now, the data doesn't have great time resolution, so it could have taken 100 million years, even a couple of 100 million years, but as far as we can tell, it happened in very much, more than an order of magnitude less than the age of the Earth, quite quickly, and consistent with very quickly indeed. The late heavy bombardment that is thought to have rendered the Earth sterile, if it had life before, it would have been uninhabitable up to that point, ended about 3.85 billion years ago, and there's almost indisputable evidence of life 3.5 billion years ago, and more controversial evidence going all the way back to about 3.8 billion years ago. So, it looks very quick. And the life we see at 3.5 billion years, it's worth emphasizing, stromatolites primarily, were already quite advanced organisms spread over much of the surface of the Earth. So they weren't the first organisms. They were already colonies of cooperating single-cell animals that had enough time to evolve the mechanisms for cooperation and to spread around the Earth. And they're still with us. Not so many. A thing that we'll be emphasizing a lot in this talk, and I already mentioned on the first slide, and you'll see it again, is the real reason that it's so hard to understand the origin of life is that it's so incredibly complex, this chemical system. And what is on top of that is that it's a huge jump in complexity with respect to abiotic chemistry. So we can, follow chemical systems up to some point, and they're reasonably complicated. And then there's this sort of huge jump in complexity that separates abiotic chemical systems from living chemical systems. And it's explaining that jump. One way of describing this is one of the chief intellectual goals of modern science is to assemble an account of how we arrived at the world as it is today. And we have an incredibly, actually, I think, successful account starting all the way back at 10 to the minus 36 seconds or something with inflation. Some of it's more secure than others, but we can give some account that's plausible and for which there's some evidence all the way up through baryogenesis and nucleosynthesis and, the fireball and the transition to the first structure. We're studying now the formation of the earliest objects in the universe. Obvious, of course, one of the leaders of that. And then we have the formation of galaxies we know something about, and inside galaxies of stars and protoplanetary systems. We know something about how planets formed, about how the Earth formed, about how its chemical composition came about. We can make a story. And subsequent to the origin of life, we can make a very good story of how life gradually developed under Darwinian evolution and growing complexity, and all the way up to, human history and cultures developed and all of, the complexity. John Harvard founded Harvard, or gave him the books, actually. And, how these things, which might be the smartest things in the room, came to be, and so on. We have a nice story. The biggest single gap in it is this jump. That's where we have to point and say, something happened there. And there's no obvious route, even speculative, for gradually bridging that route. I'll be emphasizing that, And I think of one of these things that the origin of life community averts its eyes from is there's also no surviving remnants or fossils. I put fossils in scare quotes because I include, but don't mean just literal, fossils in the ground. But there's no chemical systems still existing that are in this gap, that are intermediate between the complexity of biochemistry and the complexity of fully living system. So, and for almost the rest of that whole story, we have some kind of evidence. We can still see planetary systems forming. If you look at the gradual growth and complexity of life on Earth after it got started, we still have living examples of very simple systems. You can find creatures with, there's the eukaryotes and prokaryotes. There's simple genomes, complicated genomes. You can watch the development of the nervous systems from creatures that had only a few neurons up to us and so on. There's, everything else is, we either have some, or you can look at the microwave background and see evidence for that phase in the history of the universe. We have lots of evidence for most of this story, but there seems to be no empirical guidelines here. And there's also one thing that makes it so hard is that there are little or no theoretical insights into fundamental biology. It's not a first principles field. I mean, biology is like, any biologist in the room? Good. So biology is like, that's a dog. And, modern biology is like, that's a dog's genome. But if you ask biologists, if let's say that on the history of Earth that we're always 1 degree Kelvin warmer everywhere all the time in every place on Earth, how would the biology of Earth be different today? Just a small perturbation, perturbative theory, no clue, no idea. So would it make a difference or no difference? Just not even the sign of the effect. So we don't have any empirical or any theoretical really guidelines. We do have a sort of finish line. What we want to get is to a system which is subject to Darwinian evolution. And then we can, first of all, see in the fossil record, and second of all, understand how life became more complicated once you have a system that's subject to natural selection, the environment and its diversity drives it. And so the problem is to get, across this gap. I should speed up. So just to remind you quickly, I shouldn't get too slowed down here on what the conventional ideas are. Already back in the 1920s, long before actually the Urey Miller experiment, which is so often mentioned as starting this, theorists, Oparin and Haldane, had the idea that inputting low entropy, high free energy inputs like electrical discharges or sunlight into simple chemical compositions that you would expect on the early Earth might produce the amino acids. That's what Urey Miller experiment demonstrated in the lab. And then somehow these amino acids might plug themselves together and make nucleotides and, nucleic acid, and that somehow this could have led to early cells, and then the early cells and a primitive with a metabolism could somehow be subject to evolution and eventually develop into this first living cell, sometimes called LUCA for last universal common ancestor. This process is called abiogenesis, and you might think it should be called biogenesis, but they already had a word. Biogenesis in the 19th century already had a meaning. It meant the production of one life form from another, like a caterpillar turning into a butterfly. So I've forgotten the man's name. A famous British biologist of the 19th century coined the term abiogenesis for getting life started. And this is an old cartoon already around, I think, when I was an undergraduate. You've mostly probably all seen it. But this account of the history of life is full of, little steps like that. All of these places where it sort of says spontaneous and so on. And what's really been the most fundamental thing that's been added to that picture in the 1920s was in the 1980s, the idea developed of the RNA world. It was noticed that RNA, which is just a chain of nucleotides, of 4 nucleotides, has both replication properties and catalysis properties, self-catalysis. It can carry out chemical functions without being used up. So it can be an enzyme, ribozymes they're called, and it can also replicate, and it can also do protein synthesis. So the idea is there was some world here based on just RNA, because otherwise you have to invent RNA and proteins and cell walls all in one go to get a living step. So this is our best guess to fill in with something. So then this jumps are this RNA world has to discover the whole complicated information storage and replication system that uses DNA, and it also has to discover how to make proteins as chemical catalyst instead of doing it itself. But it's a little bit of an attempt to fill in that gap. One objection immediately is there's no RNA world around now. It's not left. Now, the hand wave of the origin of life people explanation for this is that once a more competent form of life developed, like DNA and protein-based life, it ate up and outcompeted the old RNA world and made it disappear. It erased it. That, I think, it's a possibility, but that hasn't happened anywhere else in the history of life. When eukaryotes came along, they didn't erase prokaryotes. When more complicated creatures evolve, usually the less complex ones survived in some niche. It's very rare, actually, that a whole step was erased, with no sign of what was left behind. So we don't have any RNA world existing now, as far as we know. But so something had to make it go away. That's where it is today. Let me call your attention to one particularly devilish detail of the world today. This is a ribosome, and as probably most of you know, a ribosome is an amazing macromolecule. It's actually built of several different proteins and several different RNA, and it is the universal factory used by every form of life on Earth to produce proteins. it reads in messenger RNAs, like one little reading block at a time, advancing them like a paper tape, for anyone that remembers what paper tapes are, reads off what amino acid is needed next to make whatever protein is encoded on this, pulls it out of the environment that's floating around, sticks it onto the chain, and extrudes the chain. And when the chain gets out, it's a chain of amino acids that wrap up and make whatever protein was encoded there. Every protein in the world is made by a ribosome. And the only way to make a ribosome that we're aware of that ever occurs is other ribosomes can do part of it, can do it. You have to have a ribosome to make a ribosome. It's literally a chicken and egg problem. And it's vastly complex and incredibly sensitive. It's very highly preserved. There's almost no mutations in this structure in even the most ancient creatures. There's some in these proteins, but very little in the sort of moving parts, the active parts, because you break it and you're dead. So it's one of the most highly preserved things, and yet very complicated. Somehow you had to do without that until you could produce it. I wanted to say a little bit about the requirements for natural selection. You will often hear the comment that what you need is a system which stores information flexibly and replicates it. But in fact, the requirement to get natural selection going, this is the finish line, is considerably more complicated than that. For example, you have to express that stored information. It has to make some difference to the organism or the chemical system. Otherwise, there's no selection on it. It's just copying some set of, some code or some nucleic acid sequence. But if there's no expression of the information that makes a difference for the functioning, there's no selection. So the information has to be expressed. It does have to be replicated. And the copies must be quite good, but not perfect. Because if they're perfect, there's no evolution. So you have to have errors. And on the other hand, the errors can't keep you from replicating. If you have some sort of system that's sort of minimally capable of replication, then very likely you change something and it's no longer capable of it. So you must get a system before you can turn on the magic engine of Darwinian evolution. You must have something that makes a few errors and it doesn't keep it from replicating. Moreover, the errors have to be stochastic or random in some sense, not deterministic, because you depend on those errors to react to the environment, to find solutions to environmental challenges. And that's very broadly, you can think in terms of the behaviors of, macroscopic animals to some changes in their environments, or some need for an enzyme that does some slightly different function inside a cell that's existing some different place. You're depending in natural selection on having explored the space of possible solutions to environmental challenges very broadly. And so you have to have random errors. And yet another thing that's often not paid too much attention to is this process takes energy. You're doing something that produces low entropy systems. So it requires energy, so you have to have metabolism. So but turn on natural selection, you have to already have metabolic processes that extract free energy from the environment and use it somehow. It has to eat more or less. Could be energy. So this system has to be self-powered and self-sustaining. So this is really what you need to get going, much more than just storing information, so it's more complicated. I won't talk much about these, but I should just to briefly mention, once you get natural selection going, say in the RNA world, there are other big jumps in complexity that you have to account for, like the RNA world has to somehow discover the DNA system, which is considerably more complicated. The nucleic acid enzymes have to discover how to make protein enzymes out of completely different set of monomers. They're made out of nucleotides. The proteins are made out of amino acids. Prokaryotic cells have to figure out how to become eukaryotes. Some of these, as we go along, are not as big a jumps. We have more idea. It's more plausible to understand how we got across those barriers. You have to divide into animals and plants. Metazoa have to develop. Metazoa are multi-celled organisms that have specialized cell functions. So you somehow have to go from cells that are self-sufficient to cells that are not capable of living except when they cooperate with other cells. Eventually you have to have a nervous system, cells that just process information, basically. So even once you turn on Darwinian evolution, there's some pretty big challenges down the road, but not as bad as just getting started. Now, I want to finally get to the improbable life scenario that I want to talk about today. I hesitate to say advocate, because you're going to hate this. Everybody hates this. I hate this. I hope it's not true, but that's what we're going to talk about today. So the idea of improbable life is that life arose via some extraordinarily improbable event that occurred on the Earth, some extreme statistical anomaly, which is only rational to contemplate as an explanation because we have reason to think that we live or might live in an infinite universe, or an infinite set of infinite universes, the multiverse. So
you're excluding a divine entity. This is not a religious stuff.
We'll come to that later. It's easy to. I'll show how some of the common answers to this have a certain flavor of religion about them, but let me leave that for now. So the idea is to take, to connect this problem to what we believe about the large-scale properties of the universe, and contemplate the possibility that this gap that I've been talking about in complexity was jumped kind of spontaneously in something just very, very unlikely. Basically, if you want to think of it in a sort of cartoon way, it's that, a sort of living cell somehow popped into existence, complete with ribosomes and everything else. You might not have to have that, you might probably don't have to have that big a jump. You could develop some intermediate system that evolves into it or something. But the idea is that the probability of abiogenesis per Hubble volume, per Hubble time, is some number that's very much less than 1. By the way, this is a typo. I meant to type 2 of those less than signs, but when I noticed the typo, I decided not to fix it, because this sort of, more conveys what I mean by extremely unlikely. It has the implication that there's no other life in the observable universe, most probably. And it also implies that we are unlikely to ever understand the origin of life on Earth, because it was just some fluke. So it's going to be very hard to figure out what it might have been. Now, this is an unappealing theory. The reason it's unappealing to most of us, there are actually people that like this, but I don't know what's wrong with them. But the reason that it's unappealing, in one sense, is it just predicts a universe that's very much less rich and complex and interesting and accessible to us than a universe in which life is common. It also means, of course, that astrobiology is a waste of time. Not exactly. I'll come back to how it isn't exactly, but that we're not going to find any extraterrestrial life, and that the people working in origin of life, aren't going to succeed in finding out how it got started. And the usual reaction to this distasteful theory is, I would say, irrational. The usual response is to say, well, okay, but there are other possibilities. And that's true. There are nothing I'm going to tell you today is compelling or absolutely proves the improbable life scenario. There are other ways of explaining everything, and we'll mention some of them. And you'll find yourself thinking that in the slides that are coming up. Well, there are some other possibilities. We don't have to believe something so unpleasant. But that's not that's not how we evaluate other ideas for the origin of life. Instead, we ask what evidence supports them, what they explain, what might disfavor them, how does it compare in terms of parsimony or Occam's razor or whatever to other alternatives, how good a hypothesis is it? So the goal for sort of the rest of today, the talk rather, not the day, is to sort of look at this improbable life, this unappealing theory, in a sort of fair way, the way we consider other theories of the origin of life, instead of saying we hate this. I'm like 66, and I kind of noticed a while back that reality isn't always, doesn't conform to my preferences perfectly. And so, as scientists, we have to contemplate things that are not, that the universe might not be the way we prefer. So let's start looking at some of the evidence for this approach. The strongest piece of evidence, and really what brings this, sort of forces us to look at this, is this what's sometimes called Lego life property of life. That is to say, the macromolecules of biochemistry are these polymers that are made of monomers, and the monomers have the property that you can plug any one of them into the other one. They sort of have chemical ends that plug into each other like Legos. So they're like letters of an alphabet in the familiar metaphor that I can spell out many words for. So the really good news, like from Yuri Milner and so on, and which created a great deal of excitement about the possibility of understanding the origin of life, is the monomers, the alphabet that that nature uses to construct these vastly variable macromolecules are fairly easily, not, the amino acids are much easier than the nucleotides, but they can be produced in fair concentration by natural processes in abiotic systems. So they're kind of the last step before we hit this huge gap that I've been describing. So we can get that far. And the fact that they occur naturally was thought to be very good news for origin of life studies, and it is in a way. But it carries with it the terrible consequence of this vast combinatoric space of possible polymers. It's as though you have invented the ASCII character set and discovered it occurs in nature. Now you can write any possible novel, sonnet, anything you want. But of course, there's a lot more non-novels and non-sonnets that can be written. It's the old monkeys typing on typewriters problem. How many monkeys do you have to have typing for, so on. And I'm not going to go through for this sophisticated an audience some calculation of how vast this space of combinatoric probabilities is. You're probably all familiar with that. But if you explore it, it gets really crazy. Like, if you covered every planet, you made an optimistic assumption about the number of planets in the universe and covered every planet with one monkey per square meter that typed, in 1 second a sonnet, and they did it for the age of the universe and so on. I once calculated this. You can get almost 2 lines of a sonnet, but somewhere once in the age of the universe, there's just too many non-sonnets. So the fact that we appear to have this huge combinatoric space to explore, and the fact that it looks like the minimal complexity for natural selection, the minimum length sonnet we have to write, or the minimum length biomolecule we have to invent to even start a little bit on getting to this goal of natural selection, is pretty substantial. We do have these 2 classes of macromolecules, and perhaps you have to solve this problem for the proteins and nucleic acids separately, but let's buy into the optimistic RNA world idea and assume that we just have to make a good nucleic acid, a good RNA that can somehow do both enzymic things and the replication, information storage part. How far do we have to go? And so the answer is pretty discouraging, because the polymers in question are huge. But typically they're at least hundreds of monomers long, and often hundreds of millions of monomers long in actual existing living creatures. The minimally functional proteins and nucleotides we can make or are aware of are about of order 100 long, but they really do very little. By the way, if you do one of those calculations with monkeys, instead you make 10 meters deep of pure nucleotides on every planet and let them recombine all the time, very rapidly for the history of the universe, you can explore the space up to lengths of about 60 or something like that. For comparison, the very smallest known genome is 1,821 base pairs long. That's 4 to the 1,821 possibilities you would have to write to get its genome. And it's a virus. It's not actually alive. It has to have a living cell. The simplest living cell we know of is 112,091 base pairs long. A human being, not that that's relevant, is about 3 billion. Now, you may say, why are you giving us so many significant figures? Why not just say about 112,000? Because this is in the exponent. 112,000 base pairs is 4 to the 91st power less space to explore than this. So if you work out the probability of putting together, and a ribosome is about 40 million base pairs long. Plus you need 30 or 50 amino acid sequences in the proteins, and that's, just to remind you, the only way to make a ribosome is from another one. So if you start working out the probabilities of this assembling itself accidentally, well, it's hard to calculate exactly, but you can easily get probabilities like 10 to the minus thousands, something like that, of it occurring. Now, that's insane, of course, to suggest that something with that lower probability actually occurred. The chance that I'm not actually standing here giving this talk and you're dreaming is much higher than 10 to the minus 1,000. The chance that everything we know is wrong is much higher than that. The only reason to take seriously such an absurdly small probability is if you get to multiply it by infinity. So this really depends on taking seriously an infinite universe. This is a little metaphor. Basically, the origin of life problem is like you have a set of Legos, you put them in a box, you shake it real hard for a while, and you open up a box, and by God, they've assembled themselves into a model of the U.S. Capitol building with, a little something happening here, an inauguration or something going on, in little car vehicles and made all like that. That's basic. This is a ribosome. All of these little streaks running through it are chains, specific chains of sequences of of these, instead of the Legos, 4 of these 5 nucleotides, or of the 23 amino acids. Proteins are even worse because they're at something to the, 23 to the some power instead of 4 to the some power. But anyway, so one piece of evidence is the difficulty of bridging this combinatoric space. Another one is Steve Benner, a distinguished chemist, has pointed out a number of problems with the standard model, which this improbable life model circumvents. And I just want to mention them briefly. The first problem he calls the Thar problem, or the de-evolution model problem. If your kid has Legos and makes, a model pirate ship, and then takes it all apart into its individual pieces and brings it to you and said, put it back together, that's a lot of work. But fortunately, what kids usually do is break the pirate ship into about 4 or 5 pieces, playing with it. And then since you have these big pieces, you can kind of stick them back together. Or if this were broken into a few pieces, it wouldn't be that hard to get it back together. So you would think that much easier in a probabilistic way than making life by starting out with the monomers and putting it together would be to break the basic macromolecules of life into a few big chunks and mixing them around and seeing if they have a much better chance of just sticking back together and starting to work again. We do that experiment all the time. every time something dies. Notice I'm wearing all black today to bring you news of when a cell dies, the macromolecules inside it start falling apart and degenerating. They don't have the self-maintenance mechanisms that are going on. And pretty soon you have something that consists of a sort of glump of parts of living macromolecules. And all they have to do is assemble themselves again, and the thing will come back to life. A sort of, biochemical stochastic resurrection. Doesn't happen. Instead, you get tar, just a black gooey mess. They stick together in other random ways. So that doesn't make it look too easy. All biology on Earth takes place in water solution, but the water is actually corrosive to proteins and nucleic acids. Biochemistry had to somehow develop a way to protect itself from water in order for these macromolecules to survive. That's why proteins fold. They have the amino acids all have a hydrophobic and a hydrophilic side, and proteins fold up to put the hydrophilic sides inside. I mean the hydrophobic side inside and hold the water away. There are other mechanisms that RNA uses. If you depend on chance production, the molecules are likely to occur at very low concentrations. So, if you want to make, something 100 monomers long by taking a piece that's 37 and a piece that's 63 monomers long, they're not likely to be formed if you form them randomly all in the same places, if they're independently produced. So it doesn't help much to form them piecewise. There is this point that people that love the RNA world often make, that RNA can do both self-catalysis, it can generate more copies of itself, controlled replication, and can carry out enzyme functions. But the replication and the self-catalysis, if you look at it in closely, what the actual sequences that do it in DNA, they're very different. So to have an RNA molecule, it's not like you can use the same strand of the same part of an RNA molecule to do both catalysis and replication. So you have to have something that's longer. You don't actually sort of get anything for free by sticking them into one molecule. And another point that's often made by advocates of the RNA world is that RNA enzymes actually begin to become enzymic, to become active at fairly short lengths, only like 30 or 40 nucleotides long, and they start doing something chemically, which is encouraging. That sounds like you might get some chemistry going at that level. What they overwhelmingly do is tear up other RNA molecules. And so it's not too helpful. In fact, if you try to grow long strands of RNA in the lab, what you run into is that it sort of saturates at lengths around 30 or 40, because you get up to that length and then they start cutting each other into pieces. So it's very hard work to get a statistical fluctuation that gives you something longer. And if you do, that's very likely one of the more common shorter ones will come along and cut it in two. So there's a barrier there. This is a relatively weak form of evidence. I don't want to say too much about it. It's more astrophysical. It's the point that there are arguments that the Earth might be an unusually suitable place for life to survive, from its place in the galaxy to the type of star we orbit, the distance from the star, the size of the planet, the fact that Jupiter is here protects us to some extent from giant impacts, that we have a large satellite to stabilize against precession and the resulting climatic instability, a magnetosphere to keep the solar wind from stripping off the atmosphere, plate tectonics to keep the planet from freezing into snowball states, enough water, but not so much that, the land is all buried under, 100 kilometers or something, so on and so forth. It may be that aside from these macromolecules, you need the improbability of life on the molecular level, that it also, on the macroscopic level, has relied on an improbable set of circumstances. Well, what does this model explain? Well, it explains the missing link of intermediate complexity. It explains why there's no fossils of biogenesis, no intermediate complexity systems. It's just imagining that you jumped over that by some extremely unlikely event. It's more or less designed to explain that or motivated by that. It explains the failure to synthesize life in the lab to date. And it explains the absence of other high profile life in the solar system. Now, some of you are thinking, well, yes, but there's other ways of explaining that. Yeah, there are. There are alternatives, but that we're trying to be fair. This is something that this model gets right. A somewhat similar set of things is the fact that we don't see, basically when we look off the Earth, we see wilderness, dead wilderness. The Fermi-Hart paradox that the galaxy hasn't been colonized indicates that there's no traveling, colonizing aliens, suggests that there appear to be no hyper-advanced alien civilizations that have engineered on an astronomical scale and turned galaxies into shopping malls or something that we could recognize at a large distance, the so-called Kardashev Type 2 and Type 3 civilizations. And the failure of SETI to date indicates there's no chatty aliens. A parsimonious explanation is there just aren't any, advanced aliens. So this would be a prediction. Note that this theory is falsifiable. We get a SETI signal. This idea is dead. Well, what do we have against the improbable life scenario? One is that life arose on Earth despite its incredibly small probability, but as we've already said, we would expect to find ourselves on a planet where life arose, or not really expect, we're required to find ourselves on a planet where life arose if we're in terms of the scenario of an improbable event in an infinite universe. One hint that sort of is against improbable life is the speed with which life arose on early Earth. If it were really improbable, you wouldn't expect it to happen so quickly, probably. It's like if you move to a town and they have a lottery there and you buy a ticket and the first day you win, that's evidence that the odds of winning aren't too bad, whereas if you have to buy a ticket every day for 50 years before you win, you might think is lower. And that does suggest a higher probability. Dave Spiegel and I did a careful Bayesian analysis of this, putting in the selection effects and the actual data on how early we know life existed. And the answer is, I have a couple of slides illustrating this, which I don't think I'll have time to go through, but almost the common sense conclusion is one data point with a selection bias for, if life didn't originate fairly early on Earth, it wouldn't have had time to evolve by now to an advanced form, indicates that while the sort of best estimate probability of life forming per unit Earth volume, per unit Earth age is not small. The best estimate from this one datum is that it's would make life common in the universe. However, it puts essentially no lower limit on the probability of abiogenesis. That, it turns out, is determined entirely by your prior. So if your prior is that, the probability of life arising per unit Earth-like planet per unit billion years or whatever is, 10 to the minus 7,000 or something, the early appearance of life on Earth actually has no problem with that. It's sort of a 2 sigma event. And it's determined entirely by your prior, not at all by the data. One thing that I think is promising for understanding the origin of life and is a theoretical direction which has some chance of one of the reasons I mentioned that understanding the origin of life is difficult is we have so little theoretical first principles insight into biology. A sort of promising thread of investigation in the moment is that of so-called self-organizing systems and the emergence of complexity. It seems that over a broad range of types of systems in which the interactions are very simple, the rules by which the system operates are very simple, over time, a broad class of these systems develop an astonishing amount of complexity. And people are studying this, self-organizing systems and the emergence of complexity, and looking for what general phenomena might underlie it. The way I think of this is that it might be a little bit about like there might be a sort of fourth law of thermodynamics. Thermodynamics is weird in the sense that if you look at derivations of, say, the second law, which is the most relevant one here, they're almost always done by analyzing a specific physical system, a steam engine or something, counting microstates and taking averages and things. But yet the result, the second law, applies to essentially everything. As far as we know, everything. It's in a way almost more fundamental than physical laws, because it applies to systems with almost any set of physical laws. And that's something we don't understand so well. Why something that's generally derived only by thinking about a specific physical system should have such generality. There's some ideas about it we don't have time for here. But you could imagine that there's some additional principle like the second law. And just as a hypothesis, imagine for complex systems, not complex systems, for systems that satisfy maybe some requirements on types of interactions and so on, the ratio of the local lowest entropy part to the local highest, or to the total entropy of the system, or the highest entropy part, increases with time. So we know that the total entropy has to increase, that's the second law, but maybe the distribution of entropies has to spread. If that were the case in a wide variety of systems, you would get localized low entropy, high complexity, high order regions in a system that was overall becoming more higher entropy, more disordered, lower structure. That appears to be what's happening in many systems that we can study, in fact, including the universe overall that went from all of us, this uniform plasma. It dumped all the entropy out into the photon field and made all these complex things that are our world. And maybe there's some general principle that must happen. If so, you don't have to rely on some incredibly improbable event. There's some general principle that will be driving the system towards high complexity, which is what we see. That would, I think, be, if we could understand such a fourth law of thermodynamics or something like that, that would remove the need for the improbable life scenario. It doesn't quite do everything, because LAWKI is life as we know it, if people don't know that acronym. This is used in the origin of life and astrobiology community. I didn't just make it up. You say LAWKI is life as we know it. Is presumably, even in the space of all possible complex systems, let's say all possible complex chemical systems, only a small subset are, the particular one that's realized here. If you had this sort of principle, you might expect other planets would have, chemical systems that are no less complicated than life here, but not be at all like what we call life. It'd be complicated in other ways. So the principle wouldn't necessarily explain everything, but it would be a big help. Let me skip over these. Those are the slides that talk about, the fact that the datum of early life on Earth doesn't tell you much. By the way, it could, if we had much better data, if we knew that life originated, 1 minute after the Earth became hospitable to life, then it would rule out this scenario. But it's just that the data aren't. It's the fact that it's one case, and the case that the limits on how quickly life had to form are not very strong. What's the merit relative to other available theories of the origin of life? I would say it's as good as any of the other on purely empirical grounds. No, better than many. And it's there's no particular reason to disfavor it that I can see compared to other ideas. And it also is worth pointing out that most other ideas for how life might have formed have the whiff of intelligent design, of religion about them. Because, like, one example, I've mentioned that, you make these polymers by plugging together the monomers in any order, and they do plug together in any order. But not every connection has the same binding energy. Some are a little more likely than others, a little more at a given temperature or whatever. So it could be that life simply, that there's a sort of deterministic path that leads to life at Talaki. That the reaction, some long, complicated series of reactions that leads to life as we know it is more likely than just exploring that space at random. That's an alternative. That's more or less what the standard origin of life model is today. They're searching for, an entrance way to that deterministic path. If that's true, it implies that the laws of chemistry, or the laws of physics from which the laws of chemistry, are consequences, kind of have life built into them, have the path to us kind of deterministically built into them. It makes the universe was sort of made for life, or that life is, a natural consequence of physical laws. So anything that isn't sort of purely stochastic has at least a little bit of a whiff of that about it. Which is kind of a puzzling thing, not necessarily religious, but then you have to wonder why. And a really great thing about this theory compared to any other origin of life theory I know is it's easily falsifiable. If we find extraterrestrial life of independent origin, if we make life in the lab, if aliens drop in on us or we get in touch with them, then this theory can go out the window, because in this model of an extreme statistical anomaly, you don't expect anything else in the observable universe. My undergraduate thesis advisor, Phil Morrison, once said, any discovery of extraterrestrial life or organisms outside the Earth transforms life from a miracle to a statistic. So that's sort of what that says. Here's a random Feynman quote to add sort of false gravitas. The conclusion is that we should be taking, that origin of life people and astrobiologists ought to regard this as a plausible model. A nice thing about it, there's a lot of not nice things about it, as I've said, a lot of reasons to dislike it. But a sort of appealing thing, it does have one aesthetically appealing property, which is that it connects life on Earth, as we know it, us, to the largest scale properties of the universe in a very, its infinite size and so on, in a very direct and intimate way. That's a sort of cool thing, I think, that two such different types of questions with which the universe is presented us might be connected in this way. Here's a slightly more relevant Feynman quote. I think I won't show that slide, so thank you.
Lots of questions. We'll start from this side.
Yes, you have to speak up. I'm a little deaf.
Okay, so this is an internal consistency check. I understood, I think, kind of, the approach you did to the RNA calculation of how unlikely it would be for RNA to, in and of itself, develop. Okay, and then you listed early on all the other hurdles you have to go through to even get to multi-celled life. If you do just the calculation, start with RNA, and get to multi-celled life, isn't that 10 to the hundreds in and of itself? And the fact that you had two infinitely small probabilities happen back to back mean you're probably missing something in the statistics?
Well, I think that was a two-part question. The first part, I would say, yes, it sounds to me like, that second jump from RNA to, multicellular life, and might also be an extremely improbable step. I haven't emphasized that because because it's much harder to calculate. I can calculate the probability of making an RNA molecule, and in some more in the classroom version of this talk, we do some calculations, but it's harder to calculate what that probability is, but it does look very unlikely. The second part of your question, I think my answer would be no, I don't agree, because of the selection bias. If there probably, in this model, there would be lots of places in the universe where some statistical fluctuation got you to an RNA world, that you would never get to the metazoan world. But we can't live in one of those. We wouldn't be here. So it's a sort of anthropic argument, or an anthropic selection effect better. Now, as soon as I say the word anthropic, that's a flag that we're talking about something we don't understand, and are grasping at straws, and that's the case. But right, the way I would put it is, a prediction of this is that there will be many places where you have made a much smaller leap in complexity that wouldn't get you all the way to the kind of life we have here, but might get you to some other, less complex, but still fairly complex. And there should be a whole lot more of those, exponentially more. Nevertheless, if I look at a light curve that has a 25 sigma peak, if each point's independent, it can be a long way to the next 21 sigma peak. And it just doesn't, if you go through the numbers, I think you conclude, if the probabilities are really 10 to the minus a big number, like hundreds, then you wouldn't expect it to be inside the horizon. Is that?
But that's also the weakness of this model, in the sense that, the sun is not the most abundant type of star, right? There are lower mass stars that are more abundant. The future is a much more likely time for us to exist than the present time, because there are stars that would live for 7 trillion years, the 10th of the mass of this. So why is it the sun that is that place that is so unlikely?
Well, it's the problem. The problem with anthropic reasoning is that it can explain anything. I once presented it to the philosophy department at Princeton, and they hated it and tore it to shreds. But one of the things that was pointed out, suppose Mr. Spock beamed down here between us, and from the Starship Enterprise, and gave you the Vulcan grip, and you're passed out, and all of that. Anthropic principle can explain that, no problem. We've observed it, so we must live in one of those very improbable universes where, by some crazy quantum fluctuation, much less likely than, the universe we've actually experienced so far, that occurred. So my best explanation, I wouldn't claim this as airtight reasoning, is we don't observe that we're circling a brown dwarf, living in the future, because since we don't observe it, that's not the statistical fluctuation we live in, even if it was more probable. There may be rare creatures. In other words, much rarer in the universe are the creatures that observe themselves circling a G-type star, going, why isn't it an M-type star? But we have to be one, because we've already observed that. So another explanation, of course, again, is, well, maybe life can actually live around M dwarfs for some reason we don't understand yet, and maybe we won't last very long, so the future isn't going to be very long, so maybe we're not that improbable. But those are two types of answers.
There were lots of questions on that. Yes.
Yes.
So it seems to me that we shouldn't worry about the later transitions in the history of life, because they took a long time, right? They took billions of years. And anyway, there was a lot of available statistics. There was a huge amount of primitive cells to give rise to the multicellular. So it seems that initial creation is the real problem, the first cell. So to be concrete, in your improbable life scenario, what actually happens? A single self-replicating primitive cell pops into existence? Is that what it is? What is it?
Well, yeah. First of all, I agree with the first part, is that the later stages, where things that look fairly challenging, or amazing happen, are much smaller barriers. We still don't understand them, and it would still be nice to understand them, but it's more plausible that, that those later stages evolve slowly, because we had a lot of cells and a lot of selective pressure. And in some cases, we can see, eukaryotes formed by taking over symbiotic prokaryotes as becoming part of their form, and so on. So I kind of agree with that. What the actual improbable event was, this model doesn't specify. Obviously, jumping all the way from a soup of nucleotides and amino acids that were formed naturally all the way to a living cell sitting there, sort of, like Mr. Spock beaming down, how did I get here and getting started? I think of that as the sort of conceptual thought, but the other way would be is to sort of buy into part of the standard model where they don't believe that Luca, the last universal common ancestor, which would be this sort of primitive cell, the first complete primitive cell that used ribosomes and all of that, they imagine that evolving from some simpler chemical systems, which perhaps, were not, they kind of jumped into some spot in the evolution. So maybe it's enough just to form one macromolecule that's 180 nucleotides long that does really well, and from that you can trace some causal path that doesn't rely on statistical anomaly to get you there. That's sort of the hope, is that there's some entry into this complex world of Loki that's much less simple, and all you have to do is get to that. And they're not imagining that you evolve directly into this, that through some system of interactions the living cell popped into existence. They're imagining that there's if you can get a ways into that complexity space, that it'll start bootstrapping along. For the reasons I gave, the number of demands that are required for natural selection and so on, I find that a little hard to believe, but that's another possibility, so that you don't have to make a full living cell, you just have to make some chemical system that will then, without statistical anomaly, evolve into a living cell. And the hope in the origin of life community is that there is some such path that starts at some simplicity, some order point that you might be able to access. And so that would be a less extreme version of that. But if we go back to the anthropic argument, we're most likely to live in a place where the jump, the statistical anomaly jump was as small as it possibly could be. But there would be some places where a living cell just popped into existence. Put that way, of course, it sounds completely crazy.
And you realize that you give a lot of ammunition to people that believe in intelligent design. This talk will be watched by...
So one of the, I'm sorry if you covered this in the first 10 minutes. I came in 10 minutes late. But if you didn't, one of the most effective sales approaches for SETI is the Drake equation. People always use that as a motivation for searching, because the numbers always end up impressively large. But there is this sort of Harris cartoon probability sitting in there, which is what was the topic of your talk today. How would you recast the motivation for SETI, given that your arguments today would suggest that the Drake equation is sort of an unrealistically childish approach to estimating, our probability for success?
Well, I didn't mention the Drake equation. If I were going to talk about this in the terms of the Drake equation, I would, they're one of the terms that, is the birth rate of stars times the number of planets per star that are in suitable positions times the probability life would arise on a suitable planet, and then other factors. I would just say, looks like one of those terms might be 10 to the minus 7,000 or something. And then you would motivate SETI by saying, I can test that hypothesis. I could, it's potentially falsifiable, as I said. The SETI people prefer to, I could put it this way, I have two models. The sort of standard model these days, especially the one used for press releases by the exoplanet community, is that give me water, free energy, and a cosmic abundance of elements, and a little patience, and you got life. You can read accounts that say, life is quite likely on this planet because, so on. So there's a sort of model. You could say we have a Bayesian prior, which has has a probability at about 1, or of order 1, and and then we have this improbable life scenario. So that Bayesian prior is about 10 to the minus big number, So SETI is an experiment that potentially could distinguish those two.
But you don't need SETI, you can just look at the atmospheres of transiting planets and look for oxygen or other...
Right, yeah. Any
form of SETI.
SETI's for intelligent life, but, right. So astrobiology can be thought of as, a way of, if you come from this point of view, it's a way to test your theory. And, whether that's as good a motive or not, I don't know.
Well, I would add, you could even, if you can image an exoplanet and even find evidence of photosynthesis, forget animal or intelligent life, then already you...
Yeah, I gave the Red Edge talk in this room a while back.
Or if you go to Mars and you find in the liquid water somebody, so that would be a direct test.
Yeah, any independent origin of life in the universe anywhere, including in some cases just in the laboratory, would be enough to falsify this.
Any other questions?
So you referred to this a couple times as an event, making it sound like there was a single complex molecule that arose and developed into life. Is that really what you mean? So the numbers that you give reflect that, or does this improbable event have to happen a bunch of times and only some fraction of those will survive to become?
Yeah, probably just making it once wouldn't work. I don't really know.
The probabilities you give reflect that, or is the probability even lower? Because
the probability is probably lower. Most of the factors if you rely on randomness, there's a whole series of factors I haven't included. It's not quite clear whether you want one incredibly improbable event or, a series of 10 individually not quite so harrowing. That sort of comes back to your question, not quite so harrowingly improbable events. They're, in an infinite universe, all these things will occur, and some will be more frequent than others, but I have not tried to estimate and doubt that I could, which channel is the most likely.
There is one more possibility that you didn't mention, which is that Loki, life as we know it, is not the original form of life. There was another form of life that became intelligent or somehow transported life to Earth, like panspermia.
Yeah.
And then it would obviously be sudden on Earth.
Right. Panspermia is a way of bridging this gap. It doesn't help completely because it had to have developed somewhere.
Right.
But it explains the sudden, or it doesn't change the argument that life is so complicated that it doesn't matter much if I give you the Earth or the whole universe. It's only if 10 to the 30 or something like that, or 22 gain in the size of your experiment, and you're up against 10 to the minus 100. So it doesn't change the basic argument, but it does explain the absence of remnants or fossils or hints of these sort of intermediate complexity chemical systems. And it's what that model has, that to count in its favor.
Well, if there are no more questions, let's thank everyone.